Welcome everyone. We were dealing with this process of that is how replication process takes place. We have seen the first step in the process of replication. The first step is nothing but activation of nucleotides. In the first step, that is when the process of DNA replication starts, the first thing which happens is the activation of the inactivated nucleotides takes place, and to activate those inactivated nucleotides there is an enzyme. That enzyme is called as phosphorylase. So that phosphorylase helps in that is activating of the inactivated DNA nucleotides by the addition of two phosphates. So when two phosphates are added to this adenosine monophosphate, guanosine monophosphate, they become adenosine triphosphate, guanosine triphosphate. So when they become that triphosphate, then that means the nucleotides have been activated. Okay. So the first step is over where activation of nucleotide has taken place, and simultaneously when activation is taking place, now the second step will also start. So what is the second step of DNA replication? That is opening of DNA helix. The second step in the process of replication is opening of DNA helix. DNA helix is a coiled structure. DNA is a coiled structure in helical manner. So it is a coiled structure. So this coil should be open. This coil should be open. Okay, so now here what happens is, if this is a DNA strand, first we will see this one. So this is one strand of DNA, this is another strand of DNA, two strands of DNA are there. Okay, this is one strand of DNA, this is another strand of DNA. Two strands of DNA are there. DNA is double stranded. Okay, they will be coiled in a helical manner. So we need to open these two, separate these two strands. We need to separate these two strands. So here what happens is, so when we open this, so this is the actual DNA. This is the actual helix part which is folded. Now these has to be separated. These has to be opened. The strands has to open. So here that will be the second step, which is nothing but opening of DNA helix. The second step is opening of DNA helix. In this, for the opening of DNA helix, we require an enzyme. That enzyme is DNA helicase. That enzyme is helicase. This helicase enzyme helps in the opening of DNA, that is opening of strands, DNA strands. Here first, for opening of this DNA, for the opening of this DNA, we require an oocyte. That is first for the opening of DNA, that when oocyte will be there. On the DNA strand, one oocyte will be there. Here this is the oocyte. Oocyte will be there. This oocyte is nothing but that is a sequence of nucleotides. A sequence of nucleotides from where DNA can open. What is the oocyte? Oocyte is nothing but a place where a few sequence of DNA, that is DNA, few sequence of DNA will be present. We have to open it from that place. Where we find that sequence on the DNA, we have a particular sequence. We have a particular sequence. That sequence is called as oocyte. That sequence is called as oocyte, and from that sequence only the DNA strand that starts to open up. The DNA strands start to open up. It won't open up from any place. So there is a particular place from where that opening of DNA strand will take place. There is a particular sequence on DNA from where opening of helix takes place. Helix will be coiled. So opening of helix takes place with the help of a special sequence. On the DNA helix, a special sequence of nucleotides will be there, and when that special nucleotide, that is the place where we find that specific nucleotides, special sequence of nucleotides, that place is called as ori site, and from that place the strand starts to open. Strand starts to open from ori site. It won't open from anywhere. So first, for opening of strands, that is ori site has to be recognized. Ori site is nothing but a sequence that is DNA sequence, special sequence which helps in the opening of the strand. So the first thing which need to be done is recognition of ori site. Where is that ori site on the DNA strand? Where is that ori site on the DNA strand? So the first thing is recognition of ori site, the recognition of ori site. For example, and here after this recognition, helicase enzyme binds at ori site. Helicase enzyme binds at ori site. For example, this is the DNA. Okay, special sequence will be there on this DNA. For example, special sequences here. Special sequences here. Now what happens here? Helicase enzyme comes. This is the ori site. This is the ori site. So from this site only, the opening will take place. Okay. So as soon as the recognition has taken place, okay, this is the ori site. As soon as it has been recognized that this is the ori site, what happens? The next thing is here helicase enzyme. Helicase enzyme comes and binds at that ori site. Here helicase enzyme comes and binds at that ori site. So first step is recognition of ori site. In the opening of DNA helix, the first thing which is done is recognition of ori site. Okay, ori site will be recognized on the whole DNA. One special sequence will be there. Where is that sequence? It will be first seen. Okay, once it is seen that this is the special place, what happens? Now at this special place, enzyme which is called as helicase, will come and bind at this site, attach. The enzyme helicase come and attaches itself to this ori site. As soon as this enzyme comes and attaches to the ori site, the opening will take place. The opening of DNA strand will take place. Okay. So helicase is the enzyme which helps in opening of the DNA helix, opening of the coil. DNA is in the coil form now. So to open that DNA, the enzyme is required is helicase. So what helicase will do? As soon as the ori site is recognized, helicase will go and attach itself to that place, that is ori site. Now helicase is the enzyme which is helping in opening. There is one more enzyme which is called as Topo Isomerase. There is one more enzyme which is called as Topo Isomerase, because what happens is DNA is in the coil form now. Even after you open it, when something is coiled, when you stretch it, okay, stretch it for some time, then again release it, what will happen? Again it will come back to its original place. There will be tension even after you open up. Okay, open up. When you stretch it, for that period of time it will be stretched only, but as soon as you release it, what will happen? It will again go and that is shrink itself. So tension will be there. So that tension needs to be released. Even after this enzyme starts now opening, if it starts opening also, it starts opening and it will open. What will happen? When it is there, it will be opened, but as soon as this is moving towards this side to opening of this strand, this part of the strand, what happens? This will become again coiled. This will become again coiled because there is tension in the strand. We also see this in this pen. At the tip of the pen, one spring will be there. If you stretch it now, it will stretch it. Till you hold it, it will be stretched. but as soon as you release it, now again it will go back to its original position. So here also it will be the same. When you open it, it will be opened up, but as this enzyme starts moving, as this enzymes, because here it starts opening, now it has to open. Yes, this has to be opened up. So as it is moving towards this place, now this was open now because when it moved to here, it was open. When this helicase moves to this place, now what happens? Again this part will become coiled. So there is always tension. To release this tension, to release the tension, so that even after opening up, the strand does not coil back again. We have to open the coil. For opening the coil, helicase enzyme is there. It is binding at ori site, and after binding it starts opening. Okay, after opening it should not again coil back. After opening it should not again coil back. So because it will be always, tension will always be there. As soon as it open, when it is removed from that place, now when the helicase moves from that place, it will be again become coiled. So to remove that tension, so that it does not coil again, to remove the tension so that it does not coil again after the helicase enzyme is moved. So one more enzyme will be there, which is called as Topo Isomerase, For example, the strand was like this. Okay, now helicase enzyme has bind to this. Now the strand has become like this. The strand has become like this after opening. After opening, the strand has become like this. Now what happens? This is open part. Now the helicase enzyme will move to this part, this coil part. It starts straightening this coil. What happens is, when it goes here and starts to straighten this coil, again this will become coiled. This will become coiled. So this should not happen. This should not happen. After it is opened, it should not again coil. So what? This enzyme, that is Topo Isomerase. Topo Isomerase is the enzyme which does not allow this DNA to coil. It will keep the DNA as it is. So here Topo enzyme will remove the tension. Now this has been opened. Helicase will move to this part. Now this part, this Topo Isomerase enzyme will come here. It will remove the tension from this one, so that it does not coil back again. Okay, so Topo Isomerase is the enzyme which releases the tension of the strand, so that it does not coil again. This Topo Isomerase, along with the single stranded stabilizing proteins, the strand has to be stabilized. It has to be kept open, straight. It should not coil again. So who will help the strand to stabilize, to be straight and not coil again? So these two things. One is Topo Isomerase enzyme, and the other is single strand stabilizing proteins. So this proteins and this enzyme helps the strand to be straight, and this helicase enzyme helps to open the DNA strand. Okay, so in the second step, after the that is when the nucleotides are activating itself, the step will start. The recognition of ori site will take place. After ori site is recognized, helicase will bind to that site. Helicase will bind to that site. After binding at the ori site, here the DNA starts to open up. Okay, and after opening up, there will be some tension in the strand of the open strand of the DNA. To remove that tension so that it does not coil back again, the enzyme Topo Isomerase and the few proteins, that is single stranded stabilizing proteins, helps in stabilizing the strand. Now DNA has opened up. So it looks like this. This was the coiled one, that is the strand of DNA. This strand has to open up. This is the strand of DNA. This strand has to open up. This is the helix part, you imagine. This is the helix part. Now that is recognition has taken place. It is opening. That strand which was together, it is separating. The strand which was together, helix which was together, now it is separating. So, it is separated because of this enzyme helicase and kept straight because of the enzyme Topo Isomerase. So, this is the helix part. Then this is the opened up part. This is the helix part, that is closed DNA strand. Now, this is the open DNA strand. It is not at once the whole DNA strand will be open. It will be open along, that is slowly it will be opened. First, at the starting it was like this totally. Now, this has been separated. This has been separated. This has been separated. Okay, till here it is separation has taken place. Now, what happens? It will further separate. It will further separate like this. It will further separate like this. Then slowly these two strands will totally separate. These two strands will totally separate. So, first when it starts opening now, here it is like this. This was the bind part. It started opening. So, this is the opening part and this is the helix part. So, it appears as, it resembles as fork. It resembles as fork. We have seen in this physics, tuning fork. So, it will be resembling as that of a fork. So, this is called as replication fork. A part of DNA is opened and a part of DNA is still coiled, helix form. So, this this resembles as that of a fork. So, this is called as replication fork. This is called as replication fork. And I said you, on the DNA strand polarity will be there. One will be 3' end, another will be, that is the start of the strand will have 3' end and end will have 5'. And they are anti-parallel. So, this will have 3' at the bottom and this will have 5' at the top. So, remember this one. So, this is the second step of DNA replication, where the strand, that is the DNA helix, is opening up. Now, coming to this third step. The third step is primer formation. The third step is primer formation. This primer formation we will see in the next video. Till now we have seen two steps of DNA replication. The first is recognition of oocyte and the second is, the first is activation of DNA nucleotides and the second is opening of DNA helix. Now, DNA has been opened up. After this opening of this DNA helix comes the third stage, which is nothing but primer formation. The third stage is nothing but primer formation. So, we will see this third step in the next video.